Hello, my name is Dea Gorka, and I'm a graduate student in Dr. Stormy Chamberlain's lab at UConn Health. I currently study Angelman Syndrome. Angelman Syndrome patients have a variety of really serious phenotypes such as ataxia, intellectual disability, absent speech, and severe seizures. So as you can imagine, this disease is pretty debilitating and extremely expensive for the families involved. The cause of Angelman syndrome is a loss of function of maternal UBE3A on chromosome 15. Chromosome 15, specifically region Q11 to Q13, undergoes imprinting. So UBE3A in this region, due to imprinting, is only expressed on the maternal allele. On the paternal allele, UBE3A is present, but it's silenced by this long non-coding RNA termed UBE3A antisense. What makes this locus even more complicated is that what I just described actually only occurs in neurons. So every other cell type in your body, you actually have UBE3A expression from both your maternal and your paternal allele. However, in neurons, you have the presence of this long non-coding RNA that silences UBE3A. So you no longer have UBE3A being expressed. So as you can imagine, if you have any mutations or deletions that lead to a loss of function of UBE3A on the maternal strand, these individuals now no longer have UBE3A protein present in their neurons. Currently, the exact mechanism of how this long non-coding RNA is regulated and how it leads to the silencing of UBE3A is not well understood. My thesis and poster currently focus on identifying the underlying mechanisms of how neuron-specific UBE3A antisense transcript is regulated in a cell type specific manner. In order to investigate this, we use Angelman syndrome patient-derived induced pluripotent stem cells and their neuronal derivatives as our model system. The reason that understanding the regulation of this long non-coding RNA is so important is because we know that its repression actually leads to the activation of the normally silenced paternal allele. This is an extremely promising therapeutic strategy for Angelman syndrome. There's currently no cure for this disorder, so hopefully better understanding this regulation and the mechanisms in this genomic locus can lead to novel therapeutic targets to be tested and hopefully lead to some alleviation of the phenotypes that these patients see. Hopefully what I've just explained to you piques some of your interest and you all take the time to take a look at my poster. Thank you, and I look forward to hopefully answering a ton of questions.